When identifying the bacterial organisms, you use various algorithms and identification schemes or flowcharts to help identify these organisms. And this is based off things like colony morphology, how it grows on the plate, the microscopic morphology, if it's gram-positive, gram-negative, cocci, rods, chains, pairs, those things. The biochemical characteristics, so these are some of the spot tests or different biochemical tests that are performed. And the most probable identification is based off, there's different variabilities within the organisms and how they metabolize or the different phenotypic type characteristics. So the organism may be the same genus and species, but there could be some slight variations as to how it reacts with some of these different test methods. There are some that have slower reactions or delayed reactions, such as they may ferment lactose a little bit slower than another organism that is the same genus and species. So maybe cultured from one person, it gives a certain characteristics, whereas it's cultured from another person, it gives a different characteristics. And the probability of positive versus negative reactions, so you kind of have to use some of those characteristics. If it's fermenting slower, then you have to base it off the probability of it actually producing a positive reaction versus a negative reaction. With bacterial cultivation, the bacterial growth is required in order to actually identify and characterize an organism. And so the purpose of the cultivation, or to grow it onto an agar plate, is to be able to actually grow the organism and isolate it, and isolate all of the organisms that are present within that sample. So, for example, with a stool culture, you're going to grow and isolate all of the organisms that should be normal flora within the GI tract, but you're also looking for anything that's pathogen. You're also going to determine which is the most likely cause of the infection and what is most likely a contaminant. So, for example, with nursing home urines, they typically tend to have a lot of organisms in it because it is harder to do a clean catch urine with a nursing home patient. So with those, you have to determine if it's something that should be worked up or not. With obtaining a sufficient bacterial growth, you need to make sure that you're looking at the clinically relevant bacteria, and this helps to allow for the identification. So with some circumstances, if the plate has many organisms, then you may need to subculture the organism that you wish to work up. So that would mean putting it onto another plate and allowing it to grow so that you can try to isolate just that organism. And also, by culturing the samples, then you can make sure that you're meeting the nutritional growth of all of the organisms that could possibly be within that sample. And so this includes the fastidious and non-fastidious organisms. So just to kind of recap, you can use liquid media, which is a broth, and to understand if growth is in it, you need to understand that you're looking for turbidity, those organisms actually growing in it. And then some cultures, some liquid media, will actually have a pH indicator in there so that once that bacteria grows, it will change the pH of that sample, and you can see that through a color change. And then of course you have solid media, which is most of what's going to be used, and this allows you to actually isolate individual colonies, and it also allows you to look at some of the morphological characteristics. And then in some cases you may use a semi-solid, and this is what would contain a little bit of agar as well as some broth media, and this allows for some of the restrictions with the oxygen within that type of media. And you also have different types of media. You have the enrichment media, the nutrient media. You have media that's selective and also differential, and then some of these medias can cover a couple of these categories. Your organisms will grow dependent on its environment, so whether it needs oxygen or carbon dioxide. It can vary with temperature requirements, pH, and it also needs to have the moisture. So those agar plates are fixed so that it allows the moisture to stay within that plate to prevent the organisms from drying out. And so these cultures are also incubated upside down so that that moisture does not come out of that agar and kind of evaporate up into the lid. So if you culture the organism and flip the plate over with the agar towards the top with the lid on the bottom, then this allows that moisture to stay within the agar itself. So in setting up a culture, you are streaking the plate to try to get isolation. So you are typically using a 4-quadrant method where you start in the first quadrant, you streak it, and then you turn the plate. You go back into that first quadrant a couple of times and streak it out. And then from there you will do another streak and then the 4th quadrant. With these, what you're looking for is it starts off with a thicker growth pattern, so you can see here at the bottom, and then it gets a little bit thinner and it thins out till you get to the point where you actually have individual colonies growing. And so if you look at this video here, you can see where the if I can get it to play here. So here in this video you can see where you start with that thicker streak and then it kind of gets streaked back into, and then you can start to get some colonies growing. And as it streaks out, you can kind of see those little lines as that plate tilts. You can identify bacteria based on a genotypic criteria. And so this is when the bacterial genome is characterized using a molecular technique. And so with this, you're looking for the presence of a specific gene, and this is a definitive identification of the organism. When you look at the phenotypic characteristics, you're looking what's observable physically. So you're looking at it microscopically, you're looking at how it stains, what it looks like under the microscope. You're also looking at it macroscopically, so the growth characteristics of it on a agar plate and how it grows with different media. You're also looking at the metabolic characteristics, so this is the growth requirements, the susceptibility testing to different antibiotics, and also some of the nutritional requirements and how it metabolizes various things. So one of the main things that we look at when we're trying to determine what an organism is we're looking at the colony morphology on the agar plate. And so with this, you're looking at things like size, you're looking at the color or the pigment of it, the shape of it. Is it elevated and raised? Does it have irregular borders? Does it have any special characteristics to it? How does it look for the surface of that colony? Does it glisten? Is it transparent, translucent? Is it dry? Is it dull? Is it opaque? Meaning it actually does have a color that you cannot see through. Does it look wet? Does it look mucoid? You're also looking to see if it changes the agar in any way. Does it hemolyze the agar? Does it change appearance due to some of the nutrients or different indicators that are in the agar? Does it pit the agar? Does it do anything different to that agar. And you can also use the odor of the organism growing on the plate to help characterize it. So when we look at colony morphology, we're looking at the colony as an individual, so not as a group. So these up here are mixed and you have several in a group. We're looking at them individually. So we're looking at the color. So you have some that are white. You have some varying sizes, but these are all pretty small. You have some that are transparent. So these are transparent. With this one here, it is 2 colonies that are side by side, but they have also did a partial lysis of the agar, of the blood that's in the agar. And so that would be a alpha hemolytic organism there, and since it has had a partial lysis, and so you can kind of see sort of a green tint halo around it. It's kind of hard to tell if these are raised or flat, but just from what I remember when I looked at this, the white colonies are raised, the hemolytic ones are, or the transparent ones are a little bit flatter. And so with looking at the elevation, depending on the organism, that helps you to determine a little bit about what organism it may be. And you're also looking to see if they have any sort of irregular borders, if they're round, if they're teeny tiny, kind of pinpoint, if they kind of do like a swirly type action. So you can kind of see that here, and we'll have an example of that here in a little bit. So you're just kind of looking at different characteristics of each individual colony. When we look at color, again, you have that hemolysis of the agar here, but these organisms are kind of grayish, transparent, and then you have some that are white. These colonies up here at the top, they all kind of have a whitish gray look to them. You have some over here that are kind of a yellow, or some people may say buttery. And with these, you can also kind of see a little bit of a raised center to them. So it gets a little bit darker in the center where you have more organisms in the center of it, so it kind of has a little peak. And then of course the rest of these kind of have more of a white look to them. You can see these colonies at the bottom pretty good, that they are a little bit raised. And so when you have different organisms, you can kind of, based off how it looks growing on the plate, kind of start narrowing down what the organism is. And you'll learn these characteristics as we get into the next course of microbiology and focus more on the bacteria. With size, you are looking to see if it's a large colony, medium, small, or pinpoint. And so here at the top we have some that are considered small. So you can see just in relation to the size of the colony on the plate. And so you're looking at them again as individual colonies, not as grouped together, how you have them kind of grouped in the center. And so this second plate here, they're a little bit larger. So these would be more of a medium-sized colony. And again, you've got the thicker area, and of course when they're kind of growing side by side, they look small, but once you get them out to themselves, they're a little bit bigger. And then it streaks out, and then you have another streak. And I can't quite tell if this is just a 3 or 4 quadrant streak. Then your next plate here, you have some large colonies. And then if you look at the video to the side, you can barely see those colonies, so you really have to tilt the plate to see them, but those are pinpoint. Next we look at the shape. So again, you have rounded shapes. So most of these over here that are kind of a little bit of a yellow tone are round. And you can also see some hemolysis here with this plate. So you have a beta hemolytic organism here. And then if you look at this other image, you have some smaller colonies that look fairly round. Picture's a little blurry, but they look fairly round. But then these larger colonies are irregular, so you can kind of see a little bit of an irregular shape to those. So here is an example of a culture that has a metallic sheen. So you can see as you move it, those colonies are very rough, but you can see sort of a reflective metal look to it. And some of these particular organisms will actually have a little bit of a color to that metallic sheen, so it'll look very iridescent looking. So here you have an organism that they said was a gram-negative. They couldn't remember what it was. It has a little bit of a different look because you can see a little bit of roughness to it, sort of dry looking. But what's unique about this, which is sometimes hard to see, but you can kind of see here in this bottom image, and this is what I want you to see, is where it's kind of pitting back down inside of itself. And so a lot of them are doing it where you kind of have the raised edges and then it kind of pits back down inside of itself. But the inside of these colonies look a little rough. And so this is kind of a unique look that you don't see very often. Alright, so here we have something a little bit interesting. So out here in this portion we have the organism that we looked at that had that metallic sheen. But here at the bottom, I want you to kind of focus on this, if I can get this to play at the same time. And I want you to notice how it kind of swarms out. So it starts in a pattern and then it kind of swarms out. So you can see how it kind of has little ripples or waves in it. And then that other organism, you really can't see that metallic sheen very well on this particular plate with this particular organism. And then this plate beside it on the McConkey, you can see that swarming as well. And so this morphology would be considered swarming. With these organisms, you have a mucoid look. And so these are the same organism, but where I mentioned earlier where you can have some variations in how the organisms respond. So if you tilt those plates, you can see how mucoid look those organisms appear. And you can see where it's also fermenting that lactose, so you have that real pretty purplish color with all of these organisms. Urine cultures are cultured in a way that allows you to determine the number of colony forming units per culture. And so you do this by using a calibrated loop. For most urines, you would use the 1 microliter calibrated loop, and you would dip that into the urine, and then the urine that is left inside that circle would be what is actually used to do the culture. If it was a suprapubic urine culture, so this would be where a needle and syringe is used to go directly into the bladder, and that would take a little bit of a larger loop, which is a 10 microliter loop. And then once you have done the special technique for performing that culture, then you would count the number of colonies that actually grew out, and then you would report that out. So for example, if that plate grew out greater than 100 colonies, then you would report out greater than 100 times 10 to the third colony forming units. And of course, this is just a regular culture with the 1 microliter loop. Or it'd be 100,000 colony forming units of the E. coli. So there are a couple of different ways that this is performed. It will depend on the laboratory that you work at as to which method they prefer. But you have the one technique is to, once you get that loop full of that urine, you would do a streak down the center, and then you would do a back and forth pattern. Some labs will actually do an additional streaking pattern across it. So it'll just depend on where you're at as to what they will actually require. And so you can see here where these organisms have grown out, and you have individual colonies. So you would actually count or do an estimate of these colonies. So if I were looking at this, I would get a picture of about 5, what they look like. So I would say 5, 10, 15, 20, 25, 30, 35. But there's many small ones there. So you're looking at greater than 100. So I wouldn't sit and count every colony. I would just kind of get an estimate. And so this is something that you will get used to over time. Most labs, however, do this technique here where the urine would actually be placed in the center in a little circle. So you would get it on your loop and you would just make a circle. Then you would get a clean loop and you would go into this and you would streak out. Same here, you would streak out and you would just keep going around this plate with a clean edge of the loop every time, or a new loop. And so with this video here, you can see as I tilt the plate where you can see some of those streak marks where you kind of get like a little indentation on that auger without cutting it. And you can see how those colonies grow within those streak marks. Some of the enzymatic tests that are performed, these will be listed in your chapter 12, I believe. And so what we do is we use these tests to kind of help differentiate one particular organism from another. And so as we get into the next course where we're focusing on these tests a little bit more, you will need to know these in a little bit more detail as far as which organisms are positive or negative for these individual tests. But for now, we're just looking at the test, what they are, what their principle is, and what's considered positive or negative. So some of the additional tests that are performed would be looking at the different metabolic pathways, and this is whether or not they perform an oxidation process or that they are able to ferment certain sugars. And you will get more detail on this as we get later into the bacteriology unit. You're also looking at the amino acid degradation and how they're able to use some of these products with air or without air. And you're also looking at how they utilize certain types of media and how they grow on those media and the characteristics that they present with.